Hi students, today we are going to study about packaging of DNA in eukaryotes. So before proceeding with this, I will first explain you what is the meaning of eukaryotes. See, eukaryotes means it is the cells where the nucleus is well developed. Well developed means what? The nucleus is with nucleolus and nuclear membrane. Means fully developed nucleus when present in the cell, those cells are called as eukaryotes. Okay, so today we are going to study packaging of DNA in eukaryotes or in eukaryotic cell. See, packaging of DNA means how the DNA is accommodated or how the DNA is packed or fitted inside the nucleus of the eukaryotic cell that we are going to study today. Okay, now what happens in eukaryotes? No, the size of typical nucleus is approximately about 10 raised to power minus 4 meter. See how small it is. Okay, but the length of DNA is, in our cell, the length of DNA is 2.2 meters. See the difference. The nucleus, the size of the nucleus is very small, that is 10 raised to power minus 4 meter, but the length of the DNA is 2.2 meter. Okay, so accommodation of such a long DNA in eukaryotic cell is nothing but it is a complex process or it is a complicated process. Now see what happens here. Inside this, now I am going to explain you about the packaging. For packaging, one protein is required. That protein is called as histone protein. Okay, for packaging of DNA, one protein is required. For that packaging, see this type of structure, this is called as packaging. Okay, so for this packaging, one protein is required, that is called as histone protein. Histone protein is positively charged. Okay, and it is made up of two types of amino acid. See, protein means naturally it is made up of amino acid. Histone protein is made up of two types of protein, LA, L for lysine, and another one is arginine. Okay, so what we are studying? We are studying packaging of DNA. For packaging of DNA, histone protein is required. This histone protein is made up of amino acid. It is always positively charged and it is made up of two types of amino acid, that is lysine and arginine. Okay, now we will first study about the histone protein only, because histone protein is playing important role in packaging. So we will first study about histone protein. Histone protein is always positively charged. It is made up of two types of amino acid, that is lysine and arginine. Now for this packaging, 8 histone proteins are required, 2 of each type. Okay, now see, for this packaging, 8 histone protein, 1, 2, 3, 4, 5, 6, 7, 8. 8 histone proteins are required and 2 of each type. Okay, 2 of each type means 2 will be H2A, this one and this one. One from upper side and one from lower side. This 2 will be H2A, this 2 will be H2B, this 2 will be H3, and these 2 will be H4. In this type, see 4 different types of histone proteins are here, 4 are present here. So total 8 histone proteins are required for packaging of DNA. Okay, then when these 8 histone proteins combine together, it forms octamer. It is visible from the diagram. When these 8 histone protein, they combine together, they form, these octa means 8, they forms, the structure is called as octamer. When 8 histone proteins combine together, it forms octamer. Now you know this is all about histone. Now you know DNA is negatively charged. You know why it is negatively charged? Because of the presence of phosphate group. Okay, so you have to keep in mind histone is always positively charged and DNA is negatively charged. DNA is negatively charged and histone protein is positively charged. Hence naturally the attraction will be there, opposite charges are there. So what happens, DNA wraps around the octamer and form nucleosome, and it completes 1 and 3/4 turn. Just see, this DNA is negatively charged, these histone proteins are positively charged, so naturally the attraction will be there. So DNA wraps this octamer, okay, this wraps this octamer, DNA as it is negatively charged, it wrap the octamer, and this type of structure, when there is wrapping of DNA, when there is wrapping of DNA towards the or attraction of DNA towards this nucleosome, or you can say when DNA wraps the octamer, that structure is called as nucleosome. At the time of exam, they may ask you only about nucleosome also. So what is nucleosome? Nucleosome is nothing but when DNA, negatively charged DNA, wraps the octamer of histone protein which is positively charged. That structure togetherly called as nucleosome. Okay, now next point, H1 protein is present at the position where the DNA starts wrapping and where it leaves. Okay, just see, this is green color structure, this is nothing but histone protein. This protein is called as H1 histone protein. H1 histone protein is present at the position where the DNA starts wrapping and at the place where the DNA leaves the nucleosome. Okay, so here at this position one more protein, ninth protein is present, that is called as H1 histone protein. Okay, then under electron microscope, if this structure of nucleosome is appeared or it is seen under the electron microscope, it will give the appearance of beads on string. Beads means moti. It gives the structure like beads on string. String means thread. Beads on string like structure. Now what happens now? Now see how the coiling takes place. This is how the nucleosome structure is formed. Now what happens when 6 nucleosome, see this is the single nucleosome, when these type of 6 nucleosome combines together or undergo coiling, it forms solenoid structure. When this solenoid structure again undergoes supercoiling, again undergoes coiling and coiling, it forms chromatin fibers. And when chromatin fibers undergo coiling and supercoiling, it forms chromosome. Okay, so here you can see this is the chromatin. Okay, chromatin network is present inside the nucleus. See how it is undergoing coiling. Okay, it is undergoing coiling. These are the histones. With the help of histone, they are forming nucleosome. When any nucleosome forms or combines with each other, they form solenoid structure. From solenoid, they are forming the chromosome. See how they are undergoing supercoiling and they are forming the chromosome. Okay, so now one more thing you have to keep in mind for this packaging, no, two different additional proteins are also required. H1 histone protein, H1 protein, H2A, H2B, H3, H4, these 5 types of proteins are required. But along with this, some other proteins are also required. They are called as NHCP. NHCP means non-histone chromosomal protein. So let's study about this non-histone chromosomal protein. They are of two types. Okay, one is called as heterochromatin, another one is called as euchromatin. Heterochromatin is present wherever condensed part is there. They add wherever condensed or tightly part is there, no, their heterochromatin is present. They are genetically inactive because they are too compactly they are present, no, so they are inactive, but as they are compactly present, they appears to be dark color and they contain more number of DNA because it is lengthy, no. So another type is euchromatin. It is present at non-condensed state, means wherever loose portion is there euchromatin is present. It is genetically active. It appears to be light in color, but it contains less DNA. Okay, so here we have, see here light, wherever light portion is there, their euchromatin is present, and wherever dark portion is there, tightly arranged chromosomes are there, those that part is called as heterochromatin. So here we have completed how the DNA, long DNA is packed inside the nucleus. See, long DNA is present, it undergoes coiling, super coiling takes place, and it results into the formation of chromosome. Okay, and finally it forms a chromosome and it gets fixed inside the nucleus.